All right, so today's reflex lab. We are just doing part one right now. We're gonna do the other parts, looking at all the different reflexes on Tuesday after we do the exam. So for today, we're just focusing on the first part. And why are we even interested in looking at all these reflexes? What's I mean, obviously everybody's used to the idea of, the doctor whacking on your knee with a hammer and having you kick out. Like, why is that a useful thing?
To test nerves and their response, action times.
So basically the idea of a reflex, maybe I should explain the idea of I put this in the little, hopefully everybody watched the video that I put for the intro, the idea of the reflex arc. Where you have some kind of a stimulus, integration, you figure out what to do with that information that's coming in. Typically the integration is happening in your brain or spinal cord, and some kind of a response. That's the reflex arc. And again, the response is either going to be like muscle contraction is a typical response, or it could be like glandular secretion. It's like you smell some yummy food and you start salivating or something. But this is the basic idea of a reflex arc. There's a stimulus, there's some integration, and there's a response. And a lot of the reflexes that we care about are innate reflexes. That means innate means they're just born into your nervous system. You don't have to learn them. You step on a tack and you're gonna pull your foot up really quick. You don't have to learn to pull your limbs away from a painful stimulus. That's called the withdrawal reflex, and it's just built into your nervous system. If you touch a red hot burner on the stove, you're gonna pull back without wanting to or not. You shine a light into somebody's pupil, their pupil constricts whether or not they've learned it or not. It's just innate. It's hardwired into your nervous system. So these give us a way to check if your nervous system is functioning properly, right? You check these different innate reflexes and make sure they're working properly. If they are not functioning properly, that's evidence that there's something wrong with your nervous system. There's all sorts of specific reflexes and tests you do that can look particularly at one particular nerve or another, even to see if there's damage for that nerve that would help mediate that particular reflex. So this is going to be interesting to just see them happen as they happen, but it's also clinically really useful for any kind of neurological exam, because this is how you can get information on whether or not the person's nervous system is functioning properly. So some of the ones we're going to be looking at today and on Tuesday are some of the classic reflexes people look at to assess the functioning of your nervous system. There are also learned or conditioned, I'll say versus learned or conditioned reflexes. Your nervous system is very good at kind of noticing patterns and pairing stimuli and responses. Like, example I like to give is, at this point in your life, if you're driving along and all of a sudden the brake lights come on in the car in front of you, there's not this long, like, oh, look, their lights are coming on. That must mean they're braking, which means, oh my God, their car's going to stop, which means I better stop my car. So I better brake my car before I rear end them. It's like as soon as you see their brake lights go on, your foot goes down so you don't like slam into them. You know, that's a conditioned response, that, or a conditioned reflex, I should say. That's something where you've been trained. It's a learned reflex. It's now a stimulus response, but it's not something you were born with, as opposed to the innate ones that we're going to look at in the lab today and Wednesday, which are ones that are just part of the nervous system due to its hardwiring. Other things here, the integration typically happens in the CNS, can be in the brain, could also just be in the spinal cord. The one we're looking at today, the stretch reflexes, again, hopefully you've already watched the thing that I prepared for you. They're happening just in the spinal cord. They don't even need the brain. There's a number of reflexes where the brain is not directly involved. I'll talk more about this later in lecture. There's like these kind of disturbing experiments they do with the cerebrate cats where a lot of gait reflexes and stuff, walking along or scratching, or, you know, you can actually completely obliterate all higher functionings of the brain and still have a lot of basic reflexes, like, you know, that are functioning. Like, you know, the stuff we're going to look at, this stretch reflex is just about like not falling over, right? If you start moving to one side, the muscles stretch out of there, and there's this automatic reflex that doesn't even use your brain that tightens those muscles up and keeps you upright. So any questions about the basic idea here? So I did notice that about five out of the class has nothing submitted for the pre-lab, which was a little disturbing for me because that actually is supposed to be done before we do the lab, and also is making me suspect you don't really know what the heck's going on for today's lab because you haven't read through or looked at the material, which also means you're going to be a bad lab partner because we're going to get into little groups and the other lab partners are going to know what's going on and you're going to be like, oh, what are we doing? So please, please, for yourself and for your lab partners that you end up working with in the groups, it's really important to actually do the work ahead of time. You know, and especially we're getting to a point, you know, I've been more flexible in the beginning, but we're getting to a point now where I'm going to start taking points off when things are really late, because, you know, it's just going to make things impossible to keep up in the class. So please, if you have a pre-lab that's due before the lab, you know, and we just had a half-hour break. I just double checked and some of them still weren't done. It's like, you know, instead of doing what you're doing, maybe you could have done it. So just putting that out there. Okay, so today's lab. You know, I am assuming you all already watched the initial part from the, you know, that I had up online. So let's talk about just what is the objective. What is the whole point that we're going to look at with today's lab?
To determine if stimulating the spinal cord will have an effect on the strength of the reflex.
So in what particular kind of reflexes are we looking at?
Stretch reflex.
So we're specifically looking at a couple of different stretch reflexes where the muscle gets stretched usually by tapping on a tendon attached to it, whether it's the patellar tendon attached to the quadriceps, or whether it's the Achilles tendon attached to your calf muscle, the gastrocnemius. And what is the actual little like way we're going to try to excite the spinal cord?
The what is it called? Jirasaki maneuver?
And thoracics.
Gendrasic's maneuver, Gendrasic's maneuver. It's kind of like, I don't know, when I was a kid, there were all these commercials, G.I. Joe with the Kung Fu grip. And it was like, the hands were like that and they would pull. So I can't not think about that when I think of Gendrasic's Kung Fu grip. But so basically you're pulling really hard. Sometimes people actually have you bite as well. And what is a hypothesis for how that is going to affect this stretch reflex?
I put Gendrasic's maneuver will help prevent conscious inhibition of the reflexes. It'll heighten lower limb tendon reflexes by countering some of the normal descending inhibitions the brain sends to the reflex arc.
So in terms of just independent and dependent, let's just start there. What is the relationship you're going to expect between Gendrasic's and just the magnitude of the reflex?
A positive correlation.
Yeah, so you were saying heightened, Nikki. I think it's clear also just to say like, increased magnitude or something like that. And
Like exaggerate.
yeah, and so what? And the reason why, there it really, I put that link so you could see one explanation. There are different explanations depending where you look. People have looked at this. People know it works. It's a classic part of a neurological exam. Exactly what's going on is probably complex in a number of ways. It seems like at some level, there is some level of inhibiting some inhibition, so you get more of a response. There's also a thing about just elevating overall excitatory activity in the spinal cord. So if all of your neurons are already closer to threshold because there's already a bunch of activity, then it doesn't take as much, the same stimulus is gonna probably fire off more neurons than it would have otherwise and have a stronger reaction on the muscle. Some other things talk about it just being a distracted thing. I'm more distracted doing this, so I'm not as consciously just trying to kind of hold my leg down. Probably all of those have some truth to them. So, there's more than one right answer as to why it works. Some of the people, like my educated guess is that there'll be no change. That's not a very educated guess there, because if you'd looked into it even a little, you would see everybody says that it will increase the response. So you need to do a little bit of research there to think about, what is it going to do? Especially, I gave you the link to at least one of the answers there. So how are we going to look at it? What are the two specific stretch reflexes that we're going to look at?
Patellar.
Yeah, so there's the patellar
Calcaneal.
and the calcaneal.
Okay.
So patellar, again, you've got your thigh and your shin bone and your foot bone. Patellar is, we have the quadriceps, the muscles on the top of your thigh, reach down and anchor on the top of your shin there at the tibial tuberosity. From anatomy, you're probably all used to that. Again, this is all stuff I did in much more detail on the video that's already up. And when you take the hammer and you hit there, you pull on the tendon, stretches the muscle, stretching the muscle sends a message to your spinal cord. One synapse sends a message back to the muscle and the muscle yanks. And that's it. And when the muscle yanks, your foot kicks out. Calcaneal reflex is looking at the calf muscle, which is attached to your heel bone through this tendon, the calcaneal tendon. When you hit there, it stretches this muscle, which is going to pull up here and you're going to point your toes down, plantar flexion. So those are the two stretch reflexes that we're looking at. And then in terms of the measurement, how are we going to actually measure, our dependent variable is kind of the magnitude of the reflex. But not, no, I don't want to cross my L. Magnitude of the reflex. How am I measuring the magnitude of the patellar reflex?
We're
Measuring
gonna
the angle.
calculate the angle of
Yeah.
a triangle.
Exactly. So everybody hopefully was looking at that in the, I wrote it in a lot of detail on the board. Basically, if, let's see, here's your foot. Here's your knee. You basically are going to tape a ruler onto your knee. I mean, onto your calf here. You're going to have another ruler on the floor that you can measure how many centimeters. And as you kick out, you know, this stick will go up. Let me make the stick here is here. As you kick out, the stick goes out. And what I'm really interested in is the angle here, because, you know, depending on how tall somebody is, their leg might swing out farther or less far, but I want to normalize that. So if we look at the angle, then we don't care how tall they are. We can just see the angle is larger or smaller. And to calculate this angle is just simple trigonometry. You don't need to understand the math, but you should know how to use the calculator to do the math. If this length here is B, just the distance from the pivot of your knee to the floor where you've got the ruler. If this distance is A, the distance to see how far did it actually swing out from resting. Then if I want to calculate that angle, then that angle is just going to be the inverse tan. What? Sorry about that. What the heck? There it is. It's going to be the inverse tan 10 to the minus 1 of A divided by B. A cord in this so I don't run out of juice before we're done. On your calculators. So I'm going to give you all some data in a few moments to actually figure that, to do some work on this. On your calculators, if you're doing tan, inverse tan, sometimes it's called arctan. That's a synonym for tan, inverse tan. It's the angle whose tangent is. So it's just a relationship between sides and angles. Tangent is like this sine divided by the cosine of the angle, actually. It's a ratio of the sines and the cosine. When you do this, it matters what the angle is measured in. What are the ways we can measure angles?
Degrees.
So the classic way that you're used to are degrees. You know, in which case, if I have a circle, how many degrees are in a circle?
360.
360 degrees. If I have a right angle, it's 90 degrees. That's not what you use in math. That's not what Excel uses. If you ask Excel to calculate tangent, it's not going to give you degrees or arctan. What is the units for angles when we are looking at math or science or engineering or anything like that, or statistics?
Radians.
Use radians. And radians, how many radians are in a circle?
2 pi.
2 pi, which is basically like 6.2 something or other. So really different numbers. 6 something radians in a circle, 360. So that means that it's really important you don't get these confused, which means you need to set the mode on your calculator correctly. Most calculators, not most, all calculators that do trigonometric functions let you set the mode. Usually it's degrees, radians, or there's another thing called these grads, which are for engineering. There's like 100 of these in a right angle. Don't worry about them. But you do not want radians in your calculation. So make sure when you are, I am basically going to be giving you measurements. I'll give you a measurement of what was the measurement from the pivot of their knee to the floor, give you the measurement of 3 kicks in terms of this A, and you're going to have to kind of get the average, and then you're going to have to get the angle from it. Make sure that your calculator is spitting out degrees, because we want degrees in this lab. Is that clear? What are sources of variability in this thing?
Nerve damage.
Nerve damage, for sure. What are other ones?
Amount of force in the maneuver.
Sure, how actually tight you're pulling on there. What other ones?
Any prior injuries?
Prior injuries, for sure. What are other ones?
Mathematical errors.
Yeah, that can actually that can be a huge one. If someone does not put in their calculator into degrees mode, that's going to make a big problem.
Would muscle size?
Muscle, that's more just going to be, maybe.
Mismeasurement.
So mismeasurement, the reality is it's kind of tricky to actually figure out exactly where, as you kick out, the stick is in the air. You have to use your imagination to figure out where it's going to actually hit the line here. And so the reality is these measurements are close. What about the actual just mechanics of eliciting the reflex? Every time you hit them, are you going to hit them exactly the same place, exactly the same way? Probably not. And actually everybody's response is probably even slightly different just due to whatever else in the nervous system based on what's going on in their body. How do we deal with the fact that every time you whack on below their kneecap, even if they're doing gendrastics or not, you're probably not going to get the exact same response?
I'm sorry, could you repeat that?
Say it again, Emily.
I'm sorry, could you repeat that question?
Yeah, so we know that every time you try to do the patellar tendon reflex, you hit somebody's knee and they kick out, you measure how far did they go. You're probably going to get a slightly different value. Even if nothing else has changed, you just try to do the exact same reflex, you're probably not going to get the exact same value for the angle. So how do we deal with that as a source of variability?
Calculating the average.
We do an average.
Yeah, so average of,
Like each trial.
Of the A?
Of the length.
yeah, so but you're going to do the same reflex under the same conditions more than once, right? In this case, we're going to do the exact same reflex 3 times.
Do we mark where to put it in, where to hit it in the knee?
That
So
helps.
we can,
That actually does help, but even when you do that, it's still not exactly the same every time. So,
yes.
and that's something that you're not going to experience as much because you're not getting to do it. You have to trust me, even if you know what you're doing and even if you've put a little piece of tape on their pants below their knee there, if you're thinking you're hitting exactly the same spot, you're probably not going to get the exact same kick out. So in order to deal with that randomness under the 2 conditions, we're going to do the condition where there's no gendrastics, the condition where there is gendrastics. We're going to do the same thing 3 times and then average those and then hope that number is kind of representative of like the true number. Does that make sense? So what I'm going to do now, let's see, we have 1, 2, 3. Let me see how many people we actually got in the class. We got 1, 2, 3, 4, 5, 25, 26, 27. Oh, so we got almost everybody here now. I think, yeah, we're missing 5, 26, 25, 26, 27, 28, 29, 28. Yeah, so we're missing two, I think we're just missing one. So what I'm going to do is I'm going to put you into 5 breakout rooms. It's going to just randomly figure it out. And then what I'm going to do for each of the rooms, I am going to give you some data. I'm going to give you the data that's going to tell you what is that length B. Again, you've got your lab book in there. What is that distance from the knee to the floor? And then you're also going to have 3 measurements for the kicks out, that A, that without Gendrasix, just to kind of control. And then 3 measurements for the kicks with Gendrasix. And what I would like, I'm going to have people just go into their own little rooms for 10 minutes. I want you to work on those numbers and come to a consensus in your group. You're going to come back. You should just have 2 numbers. What is the angle with Gendrasix and what is the angle without Gendrasix? So each group is going to go into a breakout room. You are going to figure out in your lab book, write down these numbers. These are like, pretend these are the measurements you got on your subject. And you're going to calculate what is the angle without Gendrasix, what is the angle with Gendrasix, and then we'll come back and we'll be able to discuss what we find. But this first thing is we're going to go into 5 breakout rooms, and it's going to be 5 to 6 people per room. It's just going to be random. I have no idea who's going where. And then once you're in there, I'm going to send messages to the different breakout rooms with your data. And then in like 10 minutes or so, we're going to all come back. So is everybody clear about what we're about to do? Actually, before we do that, I want to just show you. Let me show you the little video. I haven't shown you that one yet. This is quick. Whoop. Oh, wait, where am I? We need to get into modules. We need to get down to reflex. So this is just a few minutes, just kind of showing you what it actually looks like. All right, so we have our subject here all set up to take measurements. You can see her leg has the ruler taped onto it, and the ruler extends almost to the ground, but not quite, because obviously you want it to swing freely. And on the ground, we have a meter stick, and we want to set it up so the stick is measuring right at 0 when she's relaxed and her leg is hanging vertically. So I'll double check here, and I'm moving that so now it's at 0. So as she kicks out, like you can show as your leg kicks out, that angle of the line, you're going to have to kind of approximate where does that line meet the ruler here, and that would be this distance we're calling A. The distance from whatever pivot point to the ground, that's going to be the distance we're calling B. And then obviously you can use those two to calculate the angle. So when you're going to do this, it takes a little while. You can go up and down where her kneecap, where that tibial tuberosity is, and kind of play around. Find, oh, that wasn't the right spot exactly. That was the right spot. Yeah. You know, so you can see that, and you can put a little piece of tape and make it easier to find again. Obviously, if you go and nothing happens, don't take that data as a 0. And spend a little bit of time seeing if you can get a consistent response before you actually start getting your data. So we can do this. So right now, my subject is sitting there relaxed. We're going to get kind of our baseline measurement of the magnitude. So I'm getting ready to go. And I'm also taking a look. I'm going to just shift my position here so I'll be able to see how this goes out. So you probably don't see me here for that camera. And go. And we're going to do 3. So that went out like maybe 9 centimeters. That's how it record that. And then we're going to do it again. And we do it again. And maybe that went out like 7.5 centimeters. You don't need to get this data here. That one went out more like 16 centimeters. Here, our subject is going to do Jendrassic's maneuver. So you can see she's holding her or pulling her hands apart, and she's going to hold that tension while we do the reflex. So we're going to do this again. And now she's in Jendrassic's maneuver, coming in. And wow. So we saw there was definitely a stronger response there. That went out to maybe like 19 or so centimeters. We're going to do it one more time. Yeah, she's still pulling there. That was probably like up to 21 centimeters. We'll do one more. And that was another 19 centimeters. So there we got our data. We have 3 readings for the baseline, 3 readings for the Jendrassic's maneuver. And we're going to have to convert those into angles and get averages and see what happens.
Okay, so we're gonna test the Achilles tendon reflex, and so I will aim right here. If you want to try this at home on someone in your family, you can use your hand like kind of karate chop and get a similar reaction. And if you want to try a Jendrassic maneuver, all right. So I wasn't sure if that was larger, but maybe.
Yeah, I think that feels good more. So then Jendrassic's.
All right, Jendrassic.
Make a difference. All right, so you get a sense of what this looks like. Let me stop sharing here. So you get a sense, that's what it looks like when you're doing it. You could see just qualitatively, even when she was doing Jendrassic's, it was really kind of dramatic about how it increased things. All right, so it's, gosh darn it. You know, it's 12:25, and I feel kind of, I do want to, maybe what we should do, it might make sense to actually just finish this, just do this whole thing on Tuesday rather than do this now, because otherwise if we started now, you would not be done by 12:30, and 12:30 is the official end of the class. So I do know that people have lives beyond 12:30 that they might have scheduled in different ways. So in your lab book, be ready for the data. I don't have the data. I have not given you the data yet. You are going to get the data. You're going to analyze it. You'll get to compare it to your hypotheses. If you're just having fun at home, assuming if you're not sheltering in place alone, try to get this on somebody, and just, even if you're not measuring it quantitatively, see if you can notice it qualitatively. And again, if you don't have a official reflex hammer, you just need something to kind of whack in there, something that's kind of stiff, but not too hard that's going to hurt them. And watch what their reflex looks like when they're just sitting and hanging relaxed. Watch what happens when they're, and see what you can see. It's kind of fun to actually see it for real. And on Tuesday, we'll continue this and look at it in more detail. And if you are one of the people who does not have the pre-lab done, make sure you have that done. And otherwise, are there any comments, questions here?
When is the enzyme lab due?
When did I forgot when I made it due. Let me take a look.
Oh, you probably have it on the module.
It's on the module.
Okay, gotcha. Sorry.
Okay. Oh, and I should mention, while on that concept, a number of people have emailed me their initial analyses, wanting to make sure they're on the right track. And that's a really good thing, because for some people, I could say, yeah, you're totally on. Other people, you've got the basic idea, but in fact, maybe I should just show, I'm going to show a couple of things that people sent in to give you a sense of where you're really close, but not quite there. And it's good to, here, let's see. Like right here, I'm gonna bring this up. I'm gonna share my screen. Share. So here is somebody's graph that they're doing, and they're on the right track, right? They've got their best fit line. They've made a calculation of the slope. So that's good. But the problem is that the slope that they took is further along after things are slowing down a little. Like I added in this yellow line here saying this line here at the beginning, this more closely approximates the reaction rate when the reaction is really rolling along before it's starting to poop out here. Do you all see that? So this person is on the right track. They're doing the right thing. They got a best fit line. They're doing the calculations correct. But it'd be better if their best fit line was for the first part of the reaction when it's still going before it's starting to slow down.
So could you measure from like multiple points rather than just like one cell?
Say it again.
So on this particular example, they measured kind of like one cell. Could it be over multiple cells and calculating from there?
What do you mean cells?
Or grid spaces? They calculate from one grid space.
No, so that doesn't matter, right? That's irrelevant. If you are doing your slope correctly, if you are doing your slope from here to here or from here to here, it's the exact same slope, right?
Can you believe, doctor, I had the same answer because I took the points of the slope in the same area. So thank you.
Yeah.
I did the same mistake.
It doesn't matter where you take the two dots to get the slope from your line, because the dots aren't your data. The dots are where to get the slope of your line. I could do the slope, from here to here, or from here to here. delta y over delta x is going to give you the exact same number. There's no difference.
Exactly. So it's I took the wrong perfect fit line. That's what I probably.
Right. So let me give you another one. So here I'm going to show you one more here. Doobie-doo. So share, stop share, and new share. So here's another one. This person was thinking about having the line go through actual data points, which it doesn't have to do. In fact, usually does not go through actual data points. So what you want to do, what I showed here, again, this kind of green line that I drew, this is kind of a best fit line that is kind of approximating this graph as it's going up. And it's not necessarily going through any points. And then when you want to get the slope, you just pick any X and Y on there and you get delta X or delta Y. Doesn't even have to be, anywhere you go on this is going to be the exact same slope. So you draw your best fit line that best approximates the slope as this thing is starting to run up, and then get the slope of that line you drew, and that will be your reaction rate. So again, this person here is on the right track. They drew the line, they did their calculations correctly for that line, but the line isn't really the best fit to show the reaction rate when the reaction is really kind of getting started and moving along here. Does that make sense?
Yeah.
Yeah, I was thinking about the picking up the steepest point. So that's what I was thinking when I was doing this.
Right. So in general, more you want to just kind of approximate what's the line that fits in general, the thing there. So, and if anybody else, because this, again, I really appreciate like these people that have sent these in for feedback, because then I can just help them out there and then they end up doing it correctly and they turn it in. It's easy to grade because I can just say full credit instead of, and it's also nice because you don't end up doing a bunch of work ahead of time and then you have to redo it all. So if you like start out and it's like, am I on the right track? And I can say, yes, you keep going, or I can say the basic idea, but you want to reframe this a little bit. So, and I would definitely recommend, don't wait to do this. These people that have got it going, that's good. I think those are the people that are gonna end up being able to keep up on things. Particularly on this lab. This is a lab where every once in a while I get shocked at we're near the end of the semester and someone still hasn't turned it in, and it turns out they still haven't even started and they still haven't even actually understand what they're supposed to do. And that's not a way to pass the lab. So make sure if you're feeling intimidated by this, try to dig in. And if you find you're having trouble, ask for help.